Welcome to chapters 18 19 gene regulation. Gene regulation is basically how organisms figure out what genes, what sections of their DNA they are going to transcribe and translate to do something with them. The process is different between prokaryotes and eukaryotes, and we're going to separate them and study them independently. For practical purposes, please remember in our textbook, the gene regulation in prokaryotes, I believe, is part of chapter 18. Gene regulation in eukaryotes is part of chapter 19. So you'll have to do some magic combining those two. So let's start with bacteria. Please remember that prokaryotes, bacteria are unicellular. The cells are very, very small, and they have limited amount of space. Real estate is prime here. And since they are unicellular, the one cell needs to do absolutely everything, including sensing the environment and responding to the environment accordingly in order to survive. Now, since I mentioned that space in the cytoplasm is limited, they need to be able to switch from one activity to another fairly fast, and that is going to be a problem because bacteria do not have space to store things. Basically, whatever they have to be able to use it efficiently. So there is no storage. So you cannot say, oh, I don't need this enzyme right now. I'll just leave it sitting in my cytoplasm until later when I need it. No, because they need all the space. Now, the way bacteria regulate, before we learn a little bit about feedback inhibition, mostly the negative feedback. Let me go back to that. And if you remember, in feedback inhibition, we talked about this mostly when we did glycolysis, Krebs cycle. How do we know when to start making more ATP, when to stop? Since ATP doesn't have a long life, we just need to make it on demand. So, if you recall, feedback inhibition is you have a given pathway, and we use this with glucose and glycolysis, the process from glucose to pyruvate. You had a given number of enzymes, and if you recall, the final product can inhibit allosterically one of the enzymes early on in the cycle. That is feedback inhibition, when the product can inhibit an enzyme early on in the process, so the whole process shuts down. This can be allosteric if it attaches to a different place from the active site, or can be competitive inhibition if you are basically blocking the active site of the enzyme. So, this is what most of us do, most eukaryotic organisms. We regulate our cellular processes through feedback inhibition, just regulating the enzymes. That means, however, that if you are, I'm going to present you here with the pathway to build tryptophan. Tryptophan is one of the basic 20 amino acids that we need, and is a necessary one. We need to have it. Tryptophan is also what you find in turkey, high concentration in turkey meat, and tryptophan is the one that makes you sleepy because it's one of the precursors for dopamine. One is one of the feel-good neurotransmitters that we have in our brain. That's a side story. Anyway, so back to tryptophan. Tryptophan, the process of making tryptophan, we can make it from scratch in our cells. You have a precursor molecule, a very simple molecule, and with 5 enzymes, you end up and you can make tryptophan. However, if you are a bacteria and you do not need tryptophan, do you have space in your cells to accumulate 5 enzymes that are just sitting there? No. So, what do bacteria do? Instead of shutting down a pathway, blocking enzymes, but still keeping the enzymes there, what bacteria are going to do, they are going to block the whole process at the gene level. Basically, they are not going to produce those enzymes if they don't need them. It's a really, really cool process. So this is what we do. We block the enzymes at this level, but bacteria, prokaryotes, instead of doing it like this and saving the enzymes in their cytoplasm that are going to take space, what they are going to do is shut down the whole expression of all the genes. So they just basically do not make the enzymes at all. So they don't take space in their cytoplasm if they don't need them. The enzymes that are there, if they are not needed, they are going to be degraded by hydrolytic enzymes, and all the amino acids are going to be recycled. So, in bacteria, prokaryotic cells, instead of blocking enzyme function, you basically are going to block transcription of the genes, and you're going to regulate the whole expression of all the genes all at once. This is going to save them energy, it's going to save them space, and it's going to make the use of their cytoplasm a lot more efficient. Alright? So, back to the basics. Now, we are going to regulate transcription of the genes, process of going from DNA to RNA, in this case messenger RNA. And basically you're going to be able to turn genes on and off on demand, just like watching TV on demand. For example, we use the example of tryptophan, that is an amino acid that bacteria really need all the time. We do too, but we eat plenty. But bacteria, remember, think of the bacteria that live in our digestive system. Let's use the example of E. coli. If you have a low protein diet, you're not getting much tryptophan into your digestive system, so bacteria are going to need to use it, to do it. So usually they have that gene to make tryptophan on, and they have all the enzymes that they need working. However, if you eat enough turkey or enough protein of a given type, and you have enough tryptophan in your digestive system, do the bacteria need to keep on doing or transcription and producing all these enzymes? No. So what are they going to do? Well, I have tryptophan around, so they're going to shut down the whole process, and they're going to turn their genes off. Can they turn the genes on if they need it? Of course. So it's a on and off, but they shut down the whole expression of the genes. So basically they produce all the enzymes, or they don't produce any of the enzymes. So let's look at some specific examples and see how bacteria do this, because it's the cleverest thing. Bacteria organize all the genes that are needed for a particular process all together. They are all together. So you've seen this picture before. Here we have a DNA. And if you notice, here are a series of genes, A, B, C, D, E. These are the genes that are needed to make the 5 enzymes that are going to allow you to produce tryptophan. So usually they group all their genes together. That's what bacteria do. Genes are grouped together if they have a related function. By the way, eukaryotes, we do not do that. We have our genes all over the place in our genome. But bacteria group them together. So when they do transcription, they're going to transcribe all the genes that are needed all together, all at once. From beginning to end, everything at once. So it's pretty convenient. And these are all the genes. Now, this structure is going to have, before the gene that codes for the enzymes, is going to have what is called an operator. It's a particular sequence that is called the operator. And the operator is going to be part of another sequence that is called the promoter, just like in eukaryotic cells. Remember, the promoter is the region of the DNA that has a signal for the transcription factors to attach, and has the data box, and that's the signal basically for RNA polymerase to attach. In bacteria, you do have your promoter, specific sequence that signals, hey, here is the gene, come and attach. But this promoter also has what is called an operon, a specific sequence. So, this promoter is going to control, basically, this one promoter is going to control all the genes all at once. Let's go back to the operator. You see this little, has two little holes. Obviously, that means that something is going to be attaching to the operator. I would like for you to think of the operator as the on and off switch, like the lights, on and off switch. So, let's review very quickly. You have your DNA, you have a sequence, if you are a bacteria, you have a sequence that has all the genes that you need for a particular metabolic process. In this case, we are using the process of making tryptophan from scratch. Before the gene, you have the promoter region, that is the place where the RNA polymerase is going to attach. But this promoter region in bacteria has a particular sequence that is called the operator. And that is a place where a particular protein is going to be attaching to. Now, way before, if you keep on going up there, before you get to the gene, there is another sequence of DNA that codes for a repressor protein. Tryptophan R, this is the repressor protein. So, someplace there is something called a repressor protein. Think about what the word repressor means. To repress, to stop, to, no, you're not going to do something. So, please keep in mind that repressor protein. This group, this whole structure is what we call the operon. Most genes in bacteria are grouped together in what is called an operon, and every operon is going to have the genes that you need. You're going to have a promoter, and within the promoter, you're always going to have an operator. This whole assemblage is called an operon, and that's how genes are organized in bacteria, which is slightly different of how they are organized in eukaryotes. Now, let's talk for a second before we see the action, how this works. Let's talk about this repressor protein. So here is our operon, nice set of sequences. But before that, we usually have this other sequence of DNA that codes for a repressor protein. So this repressor protein is transcribed into messenger RNA. Messenger RNA is translated into a protein, and this is your repressor protein. Now, if you think about it, this is our operator, and now you have a protein stuck there. Nice. Look at this repressor protein here. Look at the shape. Just look at the shape of the repressor protein. Can this shape fit here? Obviously not, because this shape here means that the repressor is produced in the inactive form. As it is here, it doesn't fit here. So this repressor protein that is produced by default is always produced in the inactive form, at least in this tryptophan operon. Ooh, very scary. So, let's follow. I'm going to try to introduce you to all the players, and then we're going to see how this works. First, the metabolic pathway to produce tryptophan, since you are building a protein, or not a protein, sorry, you are building the amino acid tryptophan, that is one of the essential amino acids that you need. Since you are building tryptophan, that is going to be an anabolic process. Remember, when you build something, it's anabolism. Now, if it's an anabolic process, that means that it's something that your cell really needs. Otherwise, why are you going to be building stuff if you don't need it? So, as in general, anabolic processes are necessary and are very important, and you need them. So, if anabolic processes are needed or builds things that are needed, the gene from the very beginning, by default, is always on because you need it. So, it's always on. Electricity, we need it. So, for the most part, it's always available for us. So, anabolic processes, by default, they are usually on because they are needed. So, let's look at the operon system of an anabolic gene like tryptophan. Ready? So we have the operon, we have the sequences that code for the 5 different enzymes needed to produce tryptophan. So DNA, RNA, protein, the 5 different enzymes. Here is the operator, here is the promoter area, and here is the repressor area that produces that repressor protein. Now, let's look at this. So first, the repressor gene is producing a protein, the repressor protein, but check out the shape. Does this shape fit in there? Obviously not. So this repressor is always produced in the inactive form. It doesn't fit there. So, by default, under normal conditions, what is happening all the time? Here comes RNA polymerase, attaches to the promoter, and you have a clear path to transcribe all the genes that you need to make tryptophan. And you have a clear path because the operator is clear there. Nothing is blocking it. I know this can be kind of tricky, so let's do the noodles. So I hope you can see there. So this is, let me maybe shut down the PowerPoint. That will, no, it's not gonna work like that. Let's just leave it on. There you go. So here you have your operon system for tryptophan. You have a promoter. Here is the operator, and there you have all the genes to make tryptophan. Here at the beginning is the area where you produce the protein that is gonna be the repressor. So when things are happening, here is the repressor protein being produced by this gene. Check out the shape. Okay? So my operator that is right there, if you look, I have a couple of holes there. This obviously does not fit, or does not fit at all. So by default, this gene is always on because here comes RNA polymerase, attaches there to the promoter region, and can transcribe all the genes beautifully. That's what bacteria are always doing in your digestive system. Now let's pretend that it's Thanksgiving. You just had a massive amount of turkey, and turkey meat is high in tryptophan. Here is tryptophan. So you have a huge amount of tryptophan in your digestive system. Do the bacteria need to keep on making tryptophan from scratch? Obviously not. You have it right there in your digestive system. They are swimming in tryptophan. So what are they gonna do? They're gonna shut down the whole process. How? Well, here is the tryptophan that is in your digestive system. This tryptophan crosses the cell membrane, goes inside the cytoplasm of the bacteria, and tryptophan attaches to the repressor protein. Notice that when it attaches to the repressor protein, the shape of the protein changes. And when the shape of the protein changes, check out what happens. Fits beautifully in there. Now, plenty of tryptophan, make the repressor protein active. The repressor protein is there. So here comes RNA polymerase. Oh, let's go. What happens? It cannot do transcription. It's blocked. That's how bacteria do it. So you wait a couple of days after all the Thanksgiving leftovers are done. You go on a low protein diet, and the amount of tryptophan in your digestive system decreases. That means tryptophan goes out. Your repressor protein changes shape again, doesn't fit there anymore. So what happens now? You are ready to start making again all the enzymes that you need to make tryptophan from scratch. That's if you are a bacteria. This is the operon system, how most of their genes work. Now, before we go any further, I want to check on a couple of things. Sorry. We have this system. So, the tryptophan operon, notice that the repressor protein is produced in the inactive form, inactive, so it doesn't fit. However, when you have tryptophan present, the shape of the protein changes, become active, and is able to block that. So I want you to make a note here that in anabolic processes, the default for the gene is always on because it needs it. If the gene is going to be on, think about it, how is the repressor protein produced? If the gene is going to be on, the repressor protein has to be produced in the inactive form to make sure that it leaves the clear path across the gene. This type of operon is called a repressible operon because you can repress it. It's on by default, but if you have enough in the environment, you can repress it and stop it. Beautiful. That is a repressible operon. So anabolic pathways are usually run by what we call repressible operons. Pretty cool. We're gonna skip this. You can do it in your laptop, computer, whatever. And we're gonna go to the other type of operon system that bacteria have that works the opposite. So before we go to the next one, if you need to, please take a minute, use your textbook, look at the pictures, try to understand how this works, make a few notes, and don't memorize this at all. Please try to understand. It's all about shapes and things moving through your genes. So let's go back to bacteria now. We just talked about a system of a gene that produces enzymes for an anabolic process, something that is building something. Now, let's do the opposite. What happens with some of the catabolic processes, things that bacteria use to destroy things? Now, we're going to use the example of lactose. The main source of energy for bacteria is glucose. We have plenty, and we are using E. coli that lives in our digestive system. Very happy. Remember that we have more bacteria cells in our body than cells. Just keep that in mind. So we have E. coli. E. coli, for the most part, is using glucose because it's the most common carbohydrate that we are consuming. After we break all the complex carbs, glucose is the most common one. So that's the default pathway that they use to get energy. And usually those enzymes are always there, working full time, processing glucose. Now, what happens if you decide to go on a milk diet? Or you, for some reason, you just binge on cheese and ice cream for a while. You are going to have an excess of lactose in your digestive system. Now, what do bacteria do with lactose? Since lactose is not the most common one, carbohydrate that you have in your digestive system, usually all the enzymes needed to process lactose are off, gone, not there. But they do have the capacity to turn those genes on to produce the enzymes needed to start using lactose if glucose is not available. See, they have choices. So let's look at what happens with the group of genes for lactose. So lactose is gonna require is a energy molecule that you are gonna, or the bacteria are gonna destroy in order to produce ATP. Very similar to what happens to glucose during glycolysis. So, ready? Here we go. To break down lactose, or to be able to incorporate and use lactose, they need 3 different genes that are all together, as in all operons, in the chromosome, in the single chromosome they have. Before this, they have their operator right there, and the area where the enzyme is gonna attach. Are we ready? So, before that, again, we have the regulatory gene that is gonna produce the repressor enzyme. Now, this is a gene that they don't use all the time. So think about it. If you do not use these things, how is this gene by default? Off. So by default, this gene has to be off because they don't have lactose all the time. So if you notice, here is the regulatory gene that produces the protein. Notice the shape of the protein. The protein is produced in a nice, perfect shape that you can see how this fits there. So, the default, the regulatory protein is active, and under normal conditions, the regulatory protein is attached there. So what happens to polymerase? RNA polymerase cannot go. So the gene is off. That's what happens. So let's look at it here with my lactose operon. So here is your promoter area, your operator where the repressor protein attaches, your 3 genes there, and here is the one that is regulatory. So here is the enzyme, the protein produced, and the protein is produced in the right shape. So notice how nicely it fits there. So RNA polymerase attaches here just in case. Chung, 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 chung. It cannot transcribe anything. So under normal conditions, the gene is off, totally off. You are not producing the enzymes needed to process lactose. Now, you start eating ice cream and cheese galore. You have a lot of lactose in your digestive system, more lactose than glucose. What are bacteria gonna do? Here you have the lactose that is going through the membrane. Lactose. So what is lactose gonna do? Lactose attaches to the repressor protein, and notice what does it do to it. It changes the shape. Now the repressor protein does not fit here. Since the repressor protein does not fit here, because it has the lactose attached, what's gonna happen? Here comes RNA polymerase, attaches to the promoter area, and clear path to transcribe all the genes needed to process lactose. So all this is floating right now in the cytoplasm, and this bacteria is producing all the enzymes needed to process lactose. After 24 hours, you get sick and tired of cheese and ice cream, and you stop consuming lactose. Lactose is all used up. The repressor protein goes back to the normal shape. It fits nicely there. And your gene is back off. Pretty cool. Please focus on the visuals. This is the best way to learn about the genes. So just like what we did with the other gene, let's work on this one. Now we are talking about a catabolic gene, because it involves the breaking down of lactose. It's a catabolic, and that means that you might or might not have what you need to break in the environment. It's just an on-demand kind of thing. So the default for this gene is off. That's how the gene is usually off. When are you gonna turn it on? Only when the stuff that you need to break down, like lactose, shows up in your environment. So the default is off, and what makes it be off? You need to have something stuck there, and that's your repressor protein, but to be repressing, the protein has to be produced in the active form. This can get confusing because a lot of kind of what I call double negative things is inactive, but then it blocks, it's not, it gets confusing. But keep it straight, don't memorize, try to understand the logic and make your beautiful drawings. Now, since this gene is by default off, it's not working, this gene is called, or this operon is called an inducible operon, because you can induce it. Think of labor. People are not in labor all the time, but if you want to induce labor, you break their water and labor comes very quickly. So inducible means that you can make it come on, and this is the lactose gene right there. So again, this is a good place to stop. Please look at all the pictures and don't try to memorize. Understand the logic. Come to my room, play with the noodles, see how it works, and it's the best way to do it. But this is how bacteria regulate all their genes. They work in groups and they regulate the process of transcription of the genes. If they don't need something, they don't have any of it in the cytoplasm because they do not have the space needed to accumulate all these things. Again, I'll let you play with the animations that I have in the PowerPoint, and I think if we want to summarize this, a repressible operon usually functions in anabolic pathways. A repressible means that the operon has to be on by default and you can repress it as needed. And the opposite to that is an inducible operon that usually works in catabolic processes that you are going to be breaking down something. And basically like TV on demand. So, simple stuff, but can get confusing. Again, please do not memorize. Try to make drawings, understand it, and I hope this helps. So, this is a good place to stop, chill, process. Whenever you're ready, come back on and we'll do eukaryotic genes. Eukaryotic genes, that's us. That's what we have. And if you thought bacteria were complicated, get ready. I want to bring your attention to this beautiful picture here. Remember, we have the chromosome. The chromosome is organized in different stages, and you have the histones. Basically, the DNA loosely wraps around histones. Histones group together, and you start making ropes and loops until you form their chromosome. Most of the time, however, your DNA in your cells is somewhere between this stage, kind of loosey, not so loosey, and this stage a lot more organized. Eukaryotic cells. We are slightly different from prokaryotic cells and organisms. First, we are multicellular. Most eukaryotic cells, or at least in our case, think humans or any other multicellular organism, you're made out of many, many cells. Eukaryotic cells are bigger. That's good. If you are multicellular, that means your body needs to coordinate signals from all over the place. That means that not a single cell can decide, oh, I want to do this, and just go for it. You have to do things taking into account what other cells are doing. The basic questions for eukaryotic cells. How do we turn our genes on and off? And please think about it. We have the same DNA technically in all the parts of our body. However, I do not have teeth in my ovaries. I do not have hair in my tongue. So Obviously, I have the gene for hair in my tongue, but the gene, thankfully, is off. In my ovaries, I still have the gene for teeth. Thankfully, is off. So certain genes are on and off in different types of cells, and the question is, how and when do they turn on and off? Very clever. Part of this has to do with development, and that's going to be another chapter coming up, so hang in there. So we're going to start looking at gene regulation. Let's go back to prokaryotes. Remember, unicellular, small, they have to respond to the environment very fast, and they don't have much space, so they have to work as a unit. That's why all the genes are together grouped in an operon. In eukaryotic cells, most eukaryotics are multicellular, and basically because of this you have to coordinate the information coming from different parts of your body. Some cells will tell you to do one thing, some other parts of your body will tell you to do something else, and each cell will have to ultimately make a decision and prioritize, kind of a triage system to figure out what to do. So this means that to regulate our genes in eukaryotic cells, there are many places where you can regulate the expression of the genes. Many places where you can stop the process of producing a protein. Many. And I recommend that you take a minute right now and write this in a big piece of paper, leave space in between so the notes can breathe, so you can add little comments to each one of these. We have basically 7 places, 7 levels where we can regulate our genes, and that's also summarized here in this nice picture that you also have it in each one of your desks. The first one has to do with the packing and unpacking of your DNA around the histones. The second has to do with the process of transcription from DNA to RNA. Third one, messenger RNA processing. You are pretty good at this. Fourth, RNA transport from the messenger RNA from the nucleus to the ribosomes. Just that little stretch is a dangerous territory. A lot of regulation happening there. Fifth place where you can do regulation is during the process of translation itself. Then, maybe you already made the protein. Guess what? That's not the end yet. Because remember that later we have protein processing. We can add glycolipids, we can modify, it has to take the final shape. So during protein processing, we can still say, whoa, I made you, I don't really need you anymore, bye-bye. So different places, protein processing. And the last one, sometimes everything is done, completed, beautiful, and then you get the, no, I really don't need you. And you get destroyed at the very last minute after all this work. But all this is because there are many different levels of regulation. Your cells have to coordinate signals from everywhere, and sometimes you are half through the process when you get the signal that says, things change, and cells are flexible enough to adapt to those conditions. So I'm going to go kind of a laundry list, start from the beginning to the end. Please stop whenever you need, process, look at the pictures, and I hope this helps. So DNA packing, that's the very first one. As you recall, DNA is wrapped around histones, and they form these structures called nucleosomes. So here are my histones, and here is my DNA wrapped around my histones. Now the interesting thing is, if my DNA is wrapped around the histones, can you do transcription of that DNA? Obviously not, because there is no way for transcription factors and RNA polymerase to attach. So in order for you to do transcription, the first thing that needs to happen is that DNA has to unravel from the histones. That's the very first thing that is gonna have to happen. So, if it's tightly packed, obviously like this, tightly packed, there is obviously no transcription, and that means that your genes are turned off. Now, how can I turn these genes on, the genes that are in this piece of DNA? Well, very simple. There is something called histone acetylation. Histone acetylation. Histone acetylation. That means you add an acetyl group, acetyl, you know, a pronunciation issue. You add an acetyl group to your histones. Ready? Here is my acetyl group. If I add this acetyl group to the histones, guess what happens? They unravel. Oops, I'm having some technical difficulties. Put it the other way. There you go. The DNA unravels. And guess what? Now the genes that are here are available for transcription. So, histone acetylation, adding an acetyl group, unravels the DNA nicely, and that means that now these genes are going to be on, available for transcription. So, histone acetylation turns certain genes on. What happens if I remove my acetyl group? Bye-bye. Your DNA wraps immediately around the histones and gets compacted there. So this is a clever system, how your genes get turned on and off. Basically, this is caused by a conformational change. Adding the acetyl group, basically the shape of the protein, the histone changes and allows for the unraveling of the DNA. Good. So now we have, that's one. There's another way. So histone acetylation turns genes on because unravels your DNA. So let's go back to my DNA. We have my DNA is unraveled here. Still not getting this right. DNA is totally unraveled. So I have a particular gene here that is ready to be transcribed. Well, sometimes you change your mind and you do not want this gene transcribed. What do you do next? Your cells can do what is called DNA methylation. That means that they are going to add a methyl group, CH3, to a cytosine, only to cytosines. So here is my methyl group. I attach this methyl group to my DNA there, and guess what happens? If here comes polymerase, RNA polymerase, trying to do transcription, guess what happens when it gets to this methyl group? Gets stuck and cannot go any further. So, DNA methylation. Is this going to turn a gene on or off? Obviously, the gene is being turned off. So, DNA methylation, adding methyl groups to the cytosines in your DNA, is going to turn genes off because transcription cannot happen. If you recall bar bodies, those are, if you are, you can never have two X chromosomes functional, only one X chromosome is functional, the other one is heavily compacted, never available. What do you think can make a gene or a whole chromosome heavily compacted, that all the DNA is totally packed? One of the things that happens is this, absolutely no acetyl groups, and the other, methyl marks all over the DNA, blocking transcription of the genes. This business of adding methyl marks to your DNA to block transcription, to turn genes off, is what is called epigenetics, and it's a brand new field in biology, hot new stuff. You're gonna have to read an article and from Time magazine, simple language for everybody, that explains the basics of this. But please keep an eye on epigenetics. It's opening brand new fields, a lot of money is going there, and we'll do some work on it just to get you interested on it. So, epigenetics, basically the study of methylation in the DNA. Alright, so histone acetylation turns genes on because unravels the DNA. DNA methylation turns genes off because that methyl group blocks the path of RNA polymerase. So there you have two ways right there that have to do with regulating gene expression. Number one, so you might want to add some notes to your chart there about these two. So good place to stop and add some notes. Next one, we have transcription initiation. You know that in order to transcribe a gene, we need to have transcription factors. We need to have that promoter area, transcription factors attached to the promoter area, and in this promoter area you have the data box, thymine, adenine, that is a specific spot that says, hey, here is the beginning of the gene, RNA polymerase, come and attach here. So, that was so easy and nice. However, check this out. You have a nice picture on your desk illustrating this. Here is your promoter, again picture. Here is your promoter, here is your gene. Transcription factors and RNA polymerase attach here, and that's what you know so far. Nice, clever. However, upstream, farther up in your DNA, up there is what we call enhancer sequences. Enhancer sequences. Check it out. Up there, those are the enhancer sequences. Enhancers. Here is your gene, your promoter. There are your enhancer sequences. What happens in an enhancer sequence? Those are sequences that are recognized by other molecules, proteins, lipids, whatever they are, that attach there and are going to do a couple of things. They can activate the process of transcription to make it even better, more faster. You can be very, very fast at this, and so you have a lot more transcription, you can have more protein made, or some of the things that are attached to these enhancers can be silencer proteins. Basically, you attach there, and guess what happens? Even if you have everything else in place here, if you have a silencer there, everything is shut down. So you are ready to go here, ready to party, and here comes your parents and say, sorry, no, you're not going anywhere tonight. It's another level of control that overrides everything that happens underneath. It's pretty sad. So you have your transcription factors that attach to the promoter area, and they are ready to go, signal RNA polymerase what to attach, but to the enhancer area, you can have other things attaching that can be activator proteins or can be other molecules that make it go even higher level, faster, more of it, or silencers that kind of turn the whole thing off and shuts it down. What do you really need to remember? That there is something else up there that can increase and can decrease the expression of the gene. Promoter gene, transcription factors, all is good, but remember that there is another place where you can attach other molecules to kind of modulate the intensity of the transcription process. So that would be number 2. Alright, so now post-transcriptional control. So your gene was on, finally you are able to make it available. You are able to transcribe it. Excellent. I have a messenger RNA. Finally, I'm on my way to producing a protein. Yes. So what's going to happen now? Well, I'm gonna have to shuffle my exons and introns, and depending on how it's shuffled, which exons are joined together is the type of protein that I'm gonna make. Okay, now that's, remember, is the RNA splicing. So that's part of your post-transcriptional control. Now, after that, okay, so we have the gene is on, I was able to transcribe it, finally made messenger RNA, finally have my mature transcript after all that splicing. Now what can go wrong? Well, now we still have to add the GTP cap at the 5' end, right here, the GTP cap at the 5' end, and the poly-A tail, polyadenine tail at the 3'. And you know very well that these two components basically determine the lifespan of your messenger RNA, and that determines how much of a protein you're gonna make. A small tail, you just make a little bit. A long tail, you make a lot. So again, how much? This is all gene regulation. So the gene was on. I was able to transcribe it successfully, spliced successfully, capped it successfully, put my poly-A tail, the time machine there. How long you're gonna live? We are ready to roll. What else can go wrong? We are in our way to make our protein. There is something new up there. So your messenger RNA that is mature is on its way to the ribosome, going out. You think all is well because has the caps, so no hydrolytic enzymes can get it. Well, there is something called small interfering RNAs, siRNA. These are really short segments of RNA, usually 21 to 28 bases, that are complementary to your messenger RNA. So if I have my messenger RNA here, let's use the noodles again just for the record. I have my messenger RNA here, nicely mature segment. But then I get a piece of, good, right here. This is a small interfering RNA, basically a sequence of RNA that is complementary to this. Guess what's going to happen? If this thing that is in the cytoplasm is able to match my messenger RNA, guess what's going to happen to my messenger RNA? It's going to be destroyed. Totally destroyed. So all that work for nothing. But things happen. So basically, once you attach a small interfering RNA to a section of your messenger RNA, that is a death tag, and that messenger RNA is going to be destroyed. This is called gene silencing. Basically, you turn off the gene. That's simple. It's a tough life out there. Now, you might wonder, why all this? Why don't regulate everything at the beginning? Think about it. Remember that you're a multicellular organism. All your cells are receiving messages from all over your body, sensory, inside, think from insulin to everything, just messages everywhere. And sometimes those messages, don't tell you, are not agreeing on. So sometimes the cells have to kind of prioritize what is more important. The best analogy I have is kind of what happens to most of you. You're all very smart, motivated people. You all want to do great. You have 7 subjects to keep up with in school. And each one of us teachers think that our subject is the most important and the only one that you need to worry about, right? So AP Bio, of course, is the most important thing, and I give you all this work and I expect you to master everything. And you say yes, 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 and you do your best. But then, oh, I have a calculus quiz tomorrow. So what happens to the bio stuff? Okay, even though you started studying for bio, you have to take a break and do your calculus stuff. But then, you have a chorus performance. So all that has to be on hold because the chorus performance, you cannot put it off. Everything is like that. You have all these things that at the end of the day, you have to prioritize. All of them are needed. But you have to decide what to do. You might be on your way to study for your calculus test, but hey, you got sick. Can you study? No, so that shuts down the whole operation. So think of all these regulatory processes the same way. The cell has the best intentions, is going in one direction, when all of a sudden gets bombarded with these other things and has to stop it half the way through. So this is what we know, and there is more that we don't know that you will learn in the future as science progresses. So we had our DNA, we were able to make transcription, we escaped those small interfering RNAs, we are on our way to the ribosome. Bingo! We are ready to attach to the ribosome. Remember that the GTP, the 5' GTP, is what attaches to the ribosome there, to the small subunit, waiting for that tRNA bringing methionine so we can start the whole process. Guess what? Here's the ribosome, here is my messenger RNA, and all of a sudden, plop, this protein attaches to your messenger RNA. A protein from nowhere. Do you think the large subunit is going to be able to come on top? Obviously not. So guess what happens? Are you going to make that protein? Obviously not. So there are a number of regulatory proteins that attach to your messenger RNA when it's in the ribosome and basically prevents the large subunit from attaching, and that's the end of it. No transcript, no translation is going to be able to happen. All that work, almost made it. We are up to number 5. This is like the top 10 list. So please, if you make that list at the beginning, make sure that you make some notes as you go to clarify all the places where you can regulate the genes. Gene regulation really means, do you get to make the protein or not? And there are many places where you can stop the process. Almost there. Be patient. Finally, we have protein processing and degradation. So, oh my God, we've made the protein. You made it to the ribosome. You had the whole process of translation, and you finally have a protein. However, it's not really a protein. What is it called first? A polypeptide. Because in order to become a protein, everything needs to be folded. Sometimes some amino acids need to be trimmed. Sometimes you need to add a glyco to make a glycoprotein. You need to add carbohydrates. Sometimes you need to add lipids. Sometimes you need to add several proteins together, like in hemoglobin, so it has a quaternary structure. There are many modifications that proteins need. Sometimes you need to have a phosphate to make you active. So, you need many modifications, and until those modifications occur, you are not done yet. So, protein processing, that would be number 6. Are you folded properly? Do you have your phosphate group attached? Do you have your carbohydrates or your lipids? That's part of the protein processing. Sometimes you make the polypeptide, but they never get processed, and that's the end of it. All that work for nothing. So, finally, number 7, you made the protein. You think you're so good. You start celebrating. And then all of a sudden your body realizes, oops, too late. Things change. We don't need that protein anymore. What do you do with that protein? Well, that is what is called protein degradation. There is a beautiful protein, another protein here, called ubiquitin. Do you know what ubiquitous mean? They're everywhere. This is a funny protein. There is ubiquitin. This is your protein that is ready. You thought we're ready. Notice it's folded, twisted, ready to go. Ubiquitin is kind of a death tag. If ubiquitin attaches to you, like here, if a protein gets tagged by ubiquitin, that means you are dead, technically. You are going to be destroyed. But the ubiquitin doesn't destroy the protein, it's just tagging it. Because then comes another massive protein, the true killer here, and the true killer is called the proteasome. The words tell you the story. The proteasome is like a humongous barrel, basically surrounds the protein and basically breaks it down into the basic amino acids, destroying it. There you go. So, ubiquitin and the proteasome work together, tagging proteins that are not needed anymore in your cells, and this is how your cell knows what needs to be destroyed and recycled. Very simple. So, that's how you regulate protein synthesis. From the basic structure of DNA folding around histones to ubiquitin and proteasomes. This has been such an important process that the people that identify these small proteins called ubiquitin and the proteasomes received the Nobel Prize in 2004 for this process. Now remember, ubiquitin ends in IN. That's a clue that is a protein. So I have this. You can flip through it in PowerPoint mode. Basically, you can see all the places where RNA is processed. Good place to stop, take a deep breath, review the stuff. I just have a couple more minutes that I want you to look at this. You can also read it in your textbook as usual. But one of the common things is that what is really in our DNA? We have 3 billion base pairs, and we have about 30,000 genes, but most of our DNA actually is not coding for proteins. It's not coding for proteins. Only about 1.5% of our DNA codes for proteins. 24% is introns that are going to be removed during processing, and regulatory sequences. These are basically those sequences in the enhancers and other places that allows you to modulate the response. This is the amazing thing. About 60% of our DNA are things that repeat. Basically, they are called repetitive DNA. Does not code for protein. They are just segments that keep on repeating themselves. This is what historically has been called junk DNA, but is no longer called junk DNA. They called it junk when they didn't know what it was. Now they know that does not code for protein, but it does code for other stuff, like, for example, small interfering RNAs. Funny things. Some of the things that you're going to find there are what we call transposable elements. Those are pieces of DNA that have the capacity to move from one chromosome to another and copy themselves. Repeated sequences, that multiple copies of the same thing. This is what's called microsatellite DNA that is going to be used in DNA fingerprinting. We'll talk about it. And there are some other sections called pseudogenes. These are sequences that at some point they were true genes, but through time they have accumulated enough mutations that they are no longer functional. It's pretty amazing. These pseudogenes are really cool because allow us to trace evolutionary processes in organisms and allow us to see how one organism changes over time, because the genes are still there, but they have enough mutations that whatever they make is no longer functional. So very interesting things. The main message here is that please, only a small percentage of our DNA truly becomes protein. The rest is mostly regulatory and other stuff that we don't know, that we are discovering now. I'll let you look at transposons and retrotransposons and DNA duplication. That's very straightforward and simple, but we are gonna stop here because I think you have had enough for your brain. But please take a quick look at those other concepts towards the end of your chapter. Thank you. I hope this helped.